All right, good morning, everyone. My name is Jessica. I'm currently a research associate at the Heart BDK at UCLA. On behalf of my teammates, Gia, who is our resident O-PTM expert, Bilal, a master of machine learning, and Neo, a visiting professor of mathematics and informatics from the University of Warsaw, I would like to thank the BD2K CCC for this opportunity to present some of our recent work on ML-based integrative analytics of the oxidative stress-sensitive post-translational modification, or short O-PTM, landscape in cardiac hypertrophy. So to begin, I would like to provide some background highlighting the importance of studying cardiac hypertrophy and the role of oxidative stress PTMs that may play in the development of cardiac hypertrophy. So cardiac hypertrophy is characterized as the abnormal enlargement or thickening of the heart muscle, which often results in reduced capacity of the heart to pump blood to the tissues and organs around the body. It is recognized as a major risk factor associated with adverse outcomes in heart failure patients, which affects 5.7 million adults in the US and costs the nation $30.7 billion annually, which thus spurring research efforts to understand its underlying molecular mechanisms for developing targeted therapeutic strategies. So toward this goal, oxidative stress is becoming increasingly recognized as a key signaling event in the progression of cardiac hypertrophy. Oxidative stress describes the cellular state when free radicals in the environment outstrip the capacity of antioxidants to neutralize them. In response to oxidative stress-sensitive post-translational modifications, O-PTMs of proteins have been shown to alter the three-dimensional structure and therefore functional activities of proteins in various biological pathways, contributing to hypertrophy development. While there have been studies that associated certain O-PTM sites with cardiac remodeling, the majority of the identified O-PTM sites and proteins remain underexplored. We therefore propose a large-scale O-PTM study that would fill this knowledge gap and contribute novel mechanistic insights in the cardiac remodeling process. So in our efforts towards studying these O-PTMs, we are primarily interested in how three key features of each O-PTM site on a protein are impacted by cardiac hypertrophy. These include the modified site, or the amino acid residue which is modified, the O-PTM type, or the type of chemical modification that occurs on the amino acid site, and the occupancy, or the degree of modification of the amino acid site. And we calculate this as the percent of abundance of all sites that are modified. To date, there are at least 35 types of O-PTMs reported that span a range of amino acid residues and chemical modifications. For our study, we selected these 7 O-PTM sites representative of the spectrum of O-PTM complexity to focus on for deeper analysis of their involvement in cardiac hypertrophy. These are methionine sulfoxidation, abbreviated as MET-O, methionine sulfonation, abbreviated as MET-O2, cysteine carbonylation, abbreviated as cis-4-HNE, cysteine sulfonylation, abbreviated as cis-SO2H, cysteine sulfonylation, cis-SO3H, proline carbonylation or hydroxylation, which we abbreviate as PRO-CH, and lysine carbonylation, abbreviated as lys-2AAA. So in particular, these 7 O-PTMs are also very stable, which would also make them good candidates as biomarkers. To more comprehensively understand the interplay between O-PTMs and cardiac hypertrophy, we formulated 4 major research questions. At the most basic level, how does cardiac hypertrophy impact O-PTM features? To more accurately and precisely phenotype cardiac hypertrophy, what are the key molecular signatures or pathways contributing to hypertrophy? We also account for the temporal aspect of O-PTM regulation by asking, how does hypertrophy regulate O-PTM temporally? And finally, as the interplay between O-PTM features and other protein properties, including abundance and half-life, forms a complex regulatory network orchestrating disease progression, we pursue an integrated proteomic analytical approach that asks, how does hypertrophy comprehensively co-regulate O-PTM occupancy, protein half-life, and protein abundance? So first, I will describe the data generation and data analysis workflows for our study. To generate the data for omics analysis of O-PTM sites and proteins and hypertrophy phenotyping, left ventricle left ventricle tissues were collected from 6 genetically diverse inbred strains of mice subjected to either control or isopaternal-induced hypertrophy, abbreviated as ISO, conditions at 7 time points, days 0, 1, 3, 5, 7, 10, and 14, after the sham or ISO treatment. Proteins were then extracted and digested from these samples and underwent LC-MSMS with respect to their O-PTM features, protein abundance, and protein half-life for integrated proteomic analysis. This data generation process was largely completed and published by Edward Lau and colleagues from our lab, and links to the raw datasets can be found in their scientific data publication. For our data analysis workflow, we repurposed the raw MSMS datasets for identification and analysis of O-PTMs. We performed a search for the 7 types of O-PTMs using the Integrative Proteomics Pipeline, or IP2, developed by John Yates' group at Scripps. We were able to identify the proteins and sites modified by O-PTMs, as well as their abundance, which was used to calculate the occupancy of each O-PTM site. This O-PTM information, alongside hypertrophy phenotype information from the ISO versus control conditions, was subjected to 3 types of analytical approaches: ML-based feature selection, cubic spline-based temporal clustering, and PCA-based integrative clustering. Then, reactome pathway analysis and UniPro functional annotation were applied to each of these approaches to analyze key pathways involved in cardiac remodeling. So as illustrated in this table, the data generation and analysis workflows produced an O-PTM dataset of a total of 8,227 proteins, of which 6,521 proteins contained 25,472 sites, modified by at least one of the 7 O-PTM types of interest. Strikingly, meadow occurred on the most sites out of all 7 types, and the number of meadow sites and proteins increased following ISO, with meadow2 and ProCH exhibiting only minor increases, whereas the number of cis4HNE, cis-SO2H, cis-SO3H, and lyse2AAA actually decreased after ISO. So looking at this data, this leads us to our first research question, which is how does cardiac hypertrophy impact O-PTM features? So we first inspected how O-PTM types and associations changed following ISO treatment. Here, the circular interaction graphs portray the association between each of the 7 O-PTMs with respect to the number of their co-modified proteins during control and ISO conditions. Each node represents a type of modification, whereas each edge represents a co-modified protein. So proteins that are shared between the same 2 O-PTMs are coupled together and labeled with the total number of shared proteins in that cluster. Oops. In both control and ISO In both control and ISO conditions, we see that most of the O-PTM types are conserved. However, there are 2 cis4HNE and lyse2AA, which have been significantly decreased after ISO. And we also see highlighted in red We also see highlighted in red the types of modifications where, as illustrated in, my apologies. So we also noticed from these 2 circular graphs that O-PTMs that occur on the same amino acids tend to have closer connections or more co-modified proteins between the 2 different types of modifications. For instance, for cis4HNE and cis-SO3H, we see that there are 4 or 5 between them, which is a greater number compared to many of the other co-modified proteins with other O-PTM sites. So we next studied the effect of ISO on the distribution of O-PTM site occupancy for each O-PTM and found their occupancies to not only be widely distributed, but also distinctly varied among O-PTMs, suggesting unique dynamic regulation patterns for each O-PTM site. Notably, the average occupancy values in each O-PTM type in both control and ISO conditions all fell below 0.5, which may indicate a greater possibility for dynamic regulation of the site in question. We further analyzed the impact of ISO on the distribution of number of O-PTM sites for each type and found each to be distinct. In particular, ISO decreased the number of cis-H&E and Lys2AA modified proteins in sites per protein, as well as increased the number of meto-modified proteins in sites per protein. More than half of the modified proteins bore only one O-PTM site, even though each protein intrinsically bore numerous methionine, cysteine, proline, and lysine sites, which suggests that the O-PTM events are highly specific. So now that we examined the overall changes in O-PTM features, which are triggered by cardiac hypertrophy phenotype, we progress to our next question on what key molecular signatures or pathways may contribute to hypertrophy, or may indicate contributions to hypertrophy. So to answer this question, we employed an ML-based O-PTM feature selection and validation pipeline. First, O-PTM occupancy data sets were processed and split into training and test sets based on their genetic background. And then next, they were subjected to feature scaling for normalization following application of the feature selection and model fitting, in which the training data undergoes feature selection algorithms and a cross-validation or stability selection step, which will allow it to select features to create a predictive model. At the same time, the test data will be transformed by the selected features from that training data set to help transform the test data for the unbiased evaluation of these selected features according to the predictive model. And then from this unbiased evaluation, we are able to obtain a score of their accuracy for the prediction. So to perform ML-based feature selection, we divided our O-PTM occupancy dataset into training and testing sets based on genetic background. In more detail, this mouse family tree obtained from the Jackson Laboratory includes 102 different strains classified into 7 groups of different genetic origin, where strains in the same group have closer evolutionary distance compared to strains in different groups. So based on this tree, we can see clearly that AJ, BALB-CJ, and CEJ are the most closely related in group 1, whereas C57BL6J, DBA2J, and FVB-NJ are all in different groups and more distantly related. So based on these relationships, we created 3 criteria for determining the data splits for training versus testing data sets. Namely, the AJ, BALB-CJ, and CEJ strains are all grouped together. At minimum, there will be 4 strains in the training set, and there will be no genetic group overlapping in training and testing sets. And these criteria are all to ensure that we perform unbiased evaluation. So from applying this criteria, we produced 6 data splits based on that genetic background. And you can see in this table that each split has a different combination of the different strains. So after determining the training and testing sets, we employed a 2-stage feature selection procedure due to the curse of dimensionality, or described as P greater than N problem, in which P is the number of sites and they greatly outnumber the number of samples. So another issue resulting from this problem is that the wrapper methods become computationally expensive, and they may also overfit to the data. So in order to address these issues, we employ a first step before actually performing the ML analysis called stability selection, in which we filter out variables that are not selected once over several subsampling iterations. So it severely reduces the number of variables that the ML algorithms need to deal with. And then the input is all the O-PTM sites, and they output the PTM sites of interest. So they help narrow it down. For the second stage, we employed SVM to extract molecular signatures. And then the input is obviously the PTM sites of interest from the previous output. And then this step outputs the set of O-PTM sites, which usually number from 2 to 5, which are the most accurate. So we consider these molecular signatures, and we next subject them to validation and explanation stage, in which we found that the most accurate of the ML methods that we tried to be random forest. And then for the interpretation of how these sites may have been selected from the ML analysis, we used a program called Lime, which I will describe in a bit more detail later. So first, here are the results of molecular signature extraction. Across the 6 data splits, we found that splits 4, 5, and 6 produce both the highest accuracy and greatest overlap in the O-PTM sites that were found across the data splits. Obviously, split 6 has the highest accuracy. And then we applied line-based interpretation to try to shed light on the black box process of how this ML analysis determined those sites. So LINE stands for Local Interpretable Model Agnostic Explanations, and this tool helps guide human interpretation of the contribution of identified sites for the correct predictions. So we first looked at 3 samples from the split 6 test data set, which covers days 3, 7, and 14 of the DBA strain from the control group. And we saw that all of these were predicted correctly. And the way to read this figure is that the prediction probabilities are at the top, and they are the original predictions made by the RF classifier. And then below, we have the weights that help determine this prediction. And we see that a lot of the correct predictions will be in the same direction as the correct prediction illustrated above, and specifically Q9DC W4, Q9D6J5, and Q8BMS1, these are the UniPro IDs, all contributed to that correct prediction, whereas P47738 and Q61941 are kind of ambiguous in how they may have contributed to these predictions. We next looked at 3 samples from the split 5 test data set, which cover days 3, 5, and 10 of the C57BL strain. And this one looks at the ISO group. So we found that actually all 4 sites here identified in the split 5 contributed positively to the correct prediction. So this one's very straightforward. And finally, we looked at 3 samples from the split 4 test set, which cover day 3 from the control group and day 7 and 10 from the ISO group. So this one has a bit more of the mix. And we found that all of them were also predicted correctly. So in this set, we found Q9CQ75, Q9D6J5, Q9DCW4. You may recognize these from some of the previous splits to have contributed positively to that correct prediction, whereas P47738 is ambiguous in this split, and it was ambiguous in previous splits as well. So in summary, by employing Lime, we validated these 6 molecular signatures to be strong candidate sites for discriminating ISO and control groups. So pathway analysis of these 6 molecular signatures were found to belong to the TCA cycle or respiratory chain pathways, the fatty acid beta oxidation, and one connexin trafficking pathway protein. So next, we investigate the finer temporal dynamics of how hypertrophy regulates O-PTM of proteins. So we applied cubic splines followed by k-mean clustering to profile the temporal dynamics of O-PTMs. Specifically, in this figure, we plot the log2 transformed occupancy ratio of the O-PTM sites in ISO versus control mice, which are averaged across the 6 genetic strains, for each of the 7 time points, and found 7 clusters of different temporal profiles visualized as strip charts fitted with cubic spline-based smoothing. So in particular, we would like to highlight cluster 5, which shows a trend of decreasing O-PTM occupancy following ISO over time. And we found that this cluster was enriched in the apoptosis and cell cycle pathway, whereas cluster 6, which shows an increasing trend, was enriched in the cytoskeleton organization. Upon closer inspection, we also noticed that the proteins in cluster 5 were predominantly components of the 1433 protein complex, and in cluster 6, the ERM protein complex. These observations will be discussed in further detail in conjunction with our findings for our final research question. How does hypertrophy comprehensively co-regulate O-PTM occupancy, protein half-life, and protein abundance? To answer this question, we implemented k-mean clustering of the overall O-PTM occupancy dataset and identified 5 integrated clusters visualized as principal component analysis figures, so PCA. The top 2 principal components, indicated as PC1 and PC2, are shown in the X and Y axes, respectively. Each color encodes a k-mean cluster for a given protein, as represented by the dots, and ellipses demarcate the locations of the clusters. So each cluster is presented in corresponding color-coded line charts with respect to their O-PTM occupancy, colored in red, protein abundance, colored in green, and protein half-life, colored in blue. So in particular, we noticed cluster A was also highly enriched in respiratory chain pathway. And for this pattern, we noted a decrease in unchanged O-PTM abundance, altered O-PTM abundance with unchanged protein abundance and unchanged protein half-life or turnover, which may indicate that these proteins are regulated more directly by O-PTMs and perhaps independently of the protein itself. And then interestingly, we found that cluster B contains a lot of the 1433 as well as ERM protein complex pathway proteins. And this particular pattern is characterized by unchanged O-PTM occupancy with increased protein abundance and decreased protein half-life, which means increased protein turnover. So kind of taking all these observations together, we found that these 2 protein complexes exhibited similar changes in occupancy and protein properties during cardiac hypertrophy, but as seen in the previous analysis, they had different temporal O-PTM dynamic trends. So we tried to examine their biological functions and activities in more detail. So notably, the 1433 protein complex proteins are involved in cytoskeleton polymerization. And we see here how by interacting with the synaptopodin protein, it can help activate it. And then it also plays a role in apoptosis, as we noted earlier in the previous analysis, in which by binding to BAD, it can inhibit apoptosis. So we suspect that through these functions, the increased protein abundance and decreased protein half-life or increased turnover may allow it to better protect cells from apoptosis, as well as contribute to the cardiac hypertrophy condition during cardiac remodeling. So we also looked at the ERM protein complex proteins and saw that their function play a role in cytoskeleton organization, specifically when the ERM protein is phosphorylated, it becomes inhibited from its ability to bind to the transmembrane protein CD44, and thus inhibits the cytoskeleton organization function. However, we also found that there were reports that phosphorylation sites, the methionine residues near the phosphorylation sites, tended to also be oxidized more frequently. So there is the possibility that the O-PTMs may play a role in offsetting that the biological effect of the phosphorylation and contribute to cardiac remodeling with the cytoskeleton organization. So to summarize, our large-scale O-PTM study uses 3 analytical methods that revealed major insights toward answering each of our research questions. So first, cardiac hypertrophy does induce changes in O-PTM features, and we notice both increases and decreases across the board in the O-PTM occupancy, O-PTM types, and O-PTM sites. For the second question about the key molecular signatures and pathways, we identified 6 high-accuracy candidate molecular signatures in 3 pathways that all correctly predicted cardiac hypertrophy. For the third question, we did find 7 distinct temporal O-PTM dynamics profiles. And finally, we found 5 integrative patterns of associations among O-PTM occupancy, protein abundance, and protein half-life. And the key point that we would like to highlight here is that by combining the analyses, we were able to reveal mechanistic insights in the 1433 and ERM protein complex function in cardiac hypertrophy. So for future directions, we were thinking that to kind of expand more on the O-PTM biology and disease understanding, we can look for the hierarchical regulation property, which we did not cover in this paper, but we have a high interest for in future studies, as one of the key features of O-PTMs that can be describes a combinatorial and sequential modification of multiple O-PTM sites. So we anticipate that computational models such as probabilistic graphical models can be implemented to perform such causal relationship analysis between the key O-PTM sites and hypertrophic progression parameters such as heart rate body rate ratio and the echocardiographic data for deep phenotyping. We also have an interest in including analysis on human proteomics data sets by applying our approach that we illustrated in this study, and hopefully that can provide more clear insights in how these O-PTMs may be applied or manifest in human cardiac hypertrophy. So we also foresee a lot of future translational potential from our findings, primarily the prioritization of key molecular signatures and pathways as targets for potential therapies and novel drugs, ML-based signature extraction pipeline, which can be applied to identify biomarkers in other conditions and diseases, and our cubic spline-based temporal clustering workflow can be applied to medical data for disease progression diagnostics and drug response monitoring. So with that, I'd like to thank Dr. Pei Ping for her guidance and support of our work. GA has really spearheaded this project with her expertise in O-PTM biology. Bilal and Neil were instrumental to the machine learning approaches we use for our analyses. And we would also like to thank Howard and Dominic for their help in acquiring and processing the O-PTM datasets. Thank you for listening to my talk, and my teammates and I will be happy to take any questions.
Hi, Jessica. Very interesting talk. Thank you. So I was just very interested in the 1433 related findings, especially since that's not only one of the most interactive proteins, not just in this situation, but in a lot of other organs in the body. It's just a very highly interactive and multifunctional protein. But since any one of these protein complexes could be a mixture of up to 7 different protein isoforms, and I know that in particular the Eta isoform has been linked to cardiac hypertrophy in the past. Okay, mostly just if you don't have expression of 1433 Eta, then that's directly linked to cardiac hypertrophy in a mouse model at the very least. I'm curious if current or planned analyses would take the multiple isoforms into account.
Yeah. So in our current data sets, we have the different isoforms listed out. I don't have it shown on this slide, but I can provide you the data later if you'd like to take a look.
Cool. Yeah. No, just very curious about that.
Yeah.
Do we have any other comments or questions for Jessica?
So I thought I would, it's Pepe. I would say a few words. I I want to say congratulations to Jessica. She's spent her gap years in our BD2K center and has just been really a strong force of pushing various projects forward. And you could appreciate that she and Gia have worked on extensively oxidative stress and ER stress proteins, first on the Wikipedia project in collaboration with Dr. Andrew Su, and they've also produced a full manuscript that's already been published in Gene. And the interest that they have accumulated by doing that wiki project really served as foundation of the synthesis of this project regarding the molecular fingerprints of the oxidative stress modification. And it has now become a major research direction in my lab. So I'm very grateful to her and to Gia to have come up with this new research direction that's now stirred up tremendous interest in the cardiovascular community as well as the proteomics community. And we've also been extremely fortunate to have Neil, to have Bilal, and Professor Wei Wang giving us guidance on all these data analysis. I do wish to say that we've never been able to benefit the way we now are with the reactome knowledge base that Henning has guided us into. And with the reactome analysis, we've recognized a metabolic phenotype of this cardiac hypertrophy that was not seen before. And you could appreciate the TCA cycle as well as the beta oxidation pathways that's generated by the reactome analysis. And as Harry has pointed out, the 1433 pathways and different isoforms, even though they were linked to cardiac hypertrophy models before, but it was never a concrete association as what it's been delineated by both Neil and Bilal. So I am very grateful. I'm very excited in this new research direction we're launching now, that given us the opportunities to much better understand cardiac disease phenotypes using these data science tools. Not sure if Jessica mind, I share the good news she has with all of you. She's now having a full scholarship going to medical school for MD PhD in Chicago. So congratulations to Jessica again.